Today I'll be presenting a study called Prenatal Stress in Rats, Effects on Plasma Corticosterone, Hippocampal Glucocorticoid Receptors, and Maze Performance. First, some background on prenatal stress. It has been found that prenatal stress, or stress felt by pregnant mother rats, can alter the stress response of the infants for their entire lives. The proposed mechanism for this alteration of the HPA axis is epigenetic. This means that gene expression, but not the underlying DNA sequence, is altered. Specifically here, maternal stress can cause methylation, a chemical process that is epigenetic, of the glucocorticoid receptor gene, which causes lower glucocorticoid receptor expression. Lower glucocorticoid receptor expression means it inhibits the negative feedback function of the HPA axis because glucocorticoid receptors are critical to this negative feedback. Thus, maternal stress in the end causes reduced ability to negatively feedback in the HPA axis and a blunted stress response. They then have higher glucocorticoid baseline levels and a delayed return to baseline after stressful events. It has been hypothesized that this is an adaptive response because unstable environments could be signaled by maternal prenatal stress. Thus, a more sensitive stress response later in life might be adaptive for these unstable environments. In addition to the effect on the prenatal on the HPA axis, prenatal stress can also cause increased aggression, cognitive impairment, and alteration of sexual behavior, especially in males. There are several sexually dimorphic results that have been found, including a few studies that found that females actually have more HPA axis alteration in response to prenatal stress, and that males might have more impaired cognitive function in response to prenatal stress. However, the literature is very complex and often contradictory, so it is hard to conclude very much from it. Thus, the motivation of this study was to look at how spatial learning and the stress response were affected by prenatal stress in male and female rats. It includes a task with a normal stress level and a high stress level. The experimenters expect that in the high stress level, there will be more of a difference between prenatally stressed rats and control rats. Pregnant mother rats were divided into two groups: a control group, which was left all to their own, and an experimental group, which was subjected to prenatal restraint stress during days 15 through 19 of pregnancy by being placed in a small plastic cylinder. The offspring of these mother rats were subjected to the Morris water maze experiment, a paradigm that Brooke also used in her presentation. It is a test of spatial learning. The rats are placed into any random quadrant of the water maze and have to find a hidden platform that stays in the same place over a 9-day period. There were two conditions. One, a 20 degrees Celsius condition, or room temperature condition, which should be comfortable and less stressful. Two, a 10 degrees Celsius condition, which is about 50 degrees Fahrenheit, or equivalent to you going for a very cold ocean swim, which can be quite stressful. Researchers collected data on the escape latency of all the rats in this experiment. The escape latency is the time taken between being dropped into the Morris water maze and finding the hidden platform. Additionally, corticosterone levels were tested. Corticosterone is a type of glucocorticoid, which means that it correlates with the body's physiological response to stress. This was tested before the swim test to determine a baseline for the rats each day over the 9 days, and at 0, 30, 60, and 120 minutes after the swim test so that they could determine what the post-stressor response was in the body of the rats. Finally, after the 9th day of the experiment, the rats were sacrificed and their hippocampal glucocorticoid receptor densities were determined. The researchers expect that prenatal stress will cause an altered HPA axis function in these rats and reduced or increased escape latencies because this would signal cognitive impairment. It would take them longer to find the hidden platforms. It's hard to know what the effect of sex will be on this because of the complexity of the research. So what did they find? The researchers found that the prenatally stressed females differed from control females in terms of their HPA axis activity in both the 10 degree and 20 degree situations. they had increased baseline glucocorticoid levels, a delayed return to baseline afterwards, and lower hippocampal glucocorticoid receptor densities. These are all expected under the normal ideas of prenatal stress's effect on the HPA axis. However, in terms of spatial learning, the prenatally stressed females did not significantly differ from control females, which is somewhat surprising. Even more surprisingly, prenatally stressed males did not have any different HPA axis activity compared to control males. However, in terms of spatial memory, they were impaired compared to control males only in the 10-degree condition. So under stressful conditions, it took the prenatally stressed males longer to find the hidden platform. This is an opposite pattern seen compared to that seen in the prenatally stressed females. Here's a graph of the escape latency versus time in days that the rats took. Obviously, all of these groups got better and better over the 9 days because the hidden platform was in the same spot and they learned to find it more quickly. All of the rats seen here are prenatally stressed ones. The shaded area represents the normal escape latencies for the control group. The only significantly different line that the researchers found here by an analysis of variance test was the prenatally stressed males in the 10-degree Celsius condition, indicated by these open white circles on top. They took significantly longer than controls to find the hidden platform. However, all of the other lines did not differ significantly from controls. However, it does look like some of them are lower than the control group, which we will discuss soon. Here's a table showing the data from this experiment. In sum, the prenatally stressed males did differ from controls in terms of their spatial learning ability measured by the escape latency. Prenatally stressed females only differed in terms of their HPA axis. This is surprising, and overall leads us to a more nuanced interpretation of how prenatal stress affects spatial learning and the stress response. There are several possible confounds in this experiment. Limitations include: 1, it has been found that for prenatally stressed females, their spatial learning ability can vary with age. All of these rats were 12 months of age when they were subjected to the Morris-Water maze test. Therefore, it's possible that we did not get spatial learning differences in the prenatally stressed females because of the age. For the males, it's possible that their glucocorticoid levels were at a ceiling before the task, meaning that there would be no way for a physiological stress response to go above this and be found afterwards. However, these results still imply that the spatial learning and the stress response as a result of prenatal stress, these happen through different mechanisms and are sexually dimorphic responses in rats. One very interesting result that the researchers did not discuss that I referenced earlier was that it does seem like some of these lines down here are actually lower than controls, which would mean that the prenatally stressed rats did better than controls in finding the hidden platform. It's possible that this represents an adaptive benefit of prenatal stress, as I mentioned earlier. However, the researchers did not find statistical significance in this lower number. Specifically, the prenatally stressed females, represented by the white triangles, seem to do better than controls, which would be interesting because if it were significant, it would be very good support for the idea that prenatal stress prepares rats to deal with a more unstable environment later in life. In conclusion, in this study, prenatal stress was found to alter the stress response in female rats and cause spatial learning impairments in male rats. Thus, we have to conclude that prenatal stress causes these two different biological reactions through different mechanisms and in a sexually dimorphic way. However, much further research is needed, especially considering that this contradicts many other studies that found that males did have HPA axis alterations, and thus we cannot take these results too much at face value.